Module 2, Genetic Variation. In this module, we'll cover the wide range of variation that's seen across the genome. As we've discussed in Module 1, chromosomal mixing assures that even within families, the genetic material of offspring will be variable from the parents. Thus, even when the mother and father are the same, as in the case for some but not all of these family members, offspring will never look exactly alike. Family members share many things. In this case, family members may share eating habits and the propensity to wear bright clothing, but which may be influenced by genes. The human genome contains a large amount of sequence, only a small portion of which is variable. DNA sequence variation results in human variation. Alterations in DNA sequence can result from numerous factors, including spontaneous replication error, aging, mutagenic agents, irradiation, inflammation, malnutrition, viruses, and other factors. When a mutation occurs in a germ cell, a sperm or an egg, it can be passed on to subsequent offspring. But when a mutation occurs in a somatic cell, like a non-germ cell, it can only affect the person in which the mutation occurs. When a mutation becomes prevalent in greater than 1% of the population, it is considered a polymorphism, not a mutation. Whether a DNA sequence mutation has an overt effect on phenotype depends on where the mutation lies. A mutation or variant in the promoter or enhancer region may prevent or promote transcription factor binding, may prevent or promote RNA polymerase binding, may produce an unstable transcription factor RNA polymerase structure, may alter, either reduce or increase gene expression, or it may have no effect. Mutation or variation in the intronic region or the untranslated regions may alter RNA splicing and processing if that occurs at a splice site. It may affect stability of the nuclear RNA, which is the first phase of transcription. It may alter the protein product if it causes a change in the splice sequence. It may promote degradation of the RNA prior to processing if it occurs in the 3' untranslated region, or it may have no effect. Mutation in the exons or coding region may alter amino acid sequence. It may produce a shortened protein product. It may produce an unstable protein product. Or it may produce a non-functional protein by either disrupting active domains or disrupting protein folding or disrupting dimer or other quaternary structure formation, or it may have no effect. So even variation in the coding region is possibly to have no effect because of the wobble position and the variability in the genetic code. Chromosomal aberrations are rare and very often result in spontaneous miscarriage. Chromosomal alterations almost always occur during cellular replication, although mutagenic agents can also cause breaks and other types of damage to the chromosomes. Chromosomal variants can include non-disjunction, translocation, duplication, insertion, and ring structure formation. Non-disjunction occurs during meiosis when chromosomes fail to separate appropriately. In the normal cell, 2 different chromosomes are shown, a small and a large chromosome. But of course, in humans, there would be 23 pairs of chromosomes. In normal meiosis, each of the haploid gametes produced at the end would have 1 copy of the small and 1 copy of the large chromosome. When non-disjunction occurs in meiosis 1, all of the daughter gametes are abnormal. In this example, 2 of the gametes have 2 copies of the long chromosome, and 2 of the gametes are missing the long chromosome. When non-disjunction occurs in meiosis 2, half of the gametes are abnormal. In this example, with either an extra copy of the long chromosome or missing the long chromosome, and half of the gametes are normal. one copy of the short and one copy of the long chromosome in each cell. An alteration in chromosomal number is called aneuploidy, and two of the most common disorders of the chromosome number are trisomy 21 or Down syndrome, which has 3 copies of chromosome 21, and Klinefelter syndrome, which results from 2 copies of the X chromosome paired with a Y chromosome. Girls with Turner syndrome have an aneuploidy where they're missing 1 copy of the X chromosome. Unlike X inactivation, which is the normal process by which one X chromosome is silenced in cells, girls with Turner syndrome have some or all of their X chromosome function disrupted. Other types of chromosomal abnormalities include deletions, where genetic material is missing, duplications, where some genetic material is duplicated, and inversions, where some of the genetic material is excised or inverted. The outcome of these chromosomal abnormalities is highly dependent upon where the breaks occur and which sequences are affected. Uniparental disomy occurs when the offspring receives all or part of the genetic material for a given chromosome from only one parent. This can occur from a type of non-disjunction in both egg and sperm that results in a normal copy number, but one in which both copies come from one parent. 2 very different disorders result from either maternal uniparental disomy of chromosome 15 or a deletion or epigenetic silencing of a small region on chromosome 15 that results in only the mother's or the father's genes being expressed. Despite the same exact region being implicated, 2 distinct syndromes, Prader-Willi or Engelmann syndrome, result depending on the origin of the genetic material. Children with Engelmann syndrome have developmental delay, ataxia, and a distinct happy disposition. Prader-Willi patients have an insatiable appetite and severe obesity and are extremely challenging to treat from a nutrition standpoint. Chromosomal translocation results when the material from one chromosome switches places or is added to another chromosome. Shown here is a balanced translocation. As long as the breakpoint does not disturb a gene or a regulatory region, a balanced translocation may have no effect at all. Ring chromosomes form in conditions that facilitate chromosomal instability, such as exposure to mutagens or X radiation. High levels of inflammation and/or malnutrition are also thought to facilitate chromosomal instability, as you will read in an upcoming research paper. Copy number variants are duplicated or deleted sequences in the genome that are highly variable from person to person. These elements can include variable number tandem repeats, or VNTRs, which are variable sequences from 10 to 100 base pairs long. They can include short tandem repeats, or STRs, which are repeated sequences from 2 to 9 base pairs long, or they can also include insertion and deletion polymorphisms, which can range from 1 to a whole lot of base pairs of sequence. These elements are fairly stably fixed throughout the genome, and their variability across individuals makes them ideal for their use in DNA fingerprinting. Throughout the genome are repeated sequences that may modify expression by altering polyadenylation or splicing or RNA editing. These transposable elements are rare sequences of DNA that can move or transpose themselves to new positions within the genome of a single cell. These are known as short interspersed transposable elements, or SINEs, long interspersed transposable elements, or LINEs, or ALU sequences. Because of their variability in location, these elements have been important for tracking evolutionary development. Single nucleotide polymorphisms, or SNPs, are single base pair changes in DNA sequence. SNPs are the most common variable element in the human genome, and there are currently more than 100 million SNPs that have been reported and validated to date. SNPs have the potential to influence disease risk, response to drugs and therapies, food preferences, and even metabolism. Variability ensures that an organism and a population will be robust. Inbreeding reduces variability and increases disease susceptibility. Think purebred dogs, who are often very, very susceptible to many kinds of diseases. The more variable a population, the stronger the population. Variability is beautiful.